Welcome back to Short Burst. Today our topic is differentiate between gram-positive and gram-negative bacteria. So the discovery of this gram staining happened in the year 1884, and it was done by Hans Christian Gram, who was a microbiologist, developed the gram stain method in 1884. Now why did he do that? In order to differentiate bacterial species into two major groups. Now we know that bacteria do exist, but there are categories of them, there are species of them. So in order to differentiate it, he did this staining into two major groups based on their cell wall characteristics. So here Gram was originally trying to develop a technique to stain bacteria in tissues sample, as he was studying the relation between bacteria and diseases, particularly in relation to pneumonia. What happened that Gram's goal was to create stain method that could differentiate bacteria, particularly those that causes disease, and allowed them to be easily observed under a microscope, because staining makes it easy to understand. The Gram stain involved applying a crystal violet dye. So the dye is actually what we are going to study, and how does it help. So followed by iodine, which binds to the dye, alcohol or acetone to wash away the dye, to finally a counter stain. Okay, so let's get one thing very easy, like there are certain definitions that will make this topic easier. So let's complete that. So the bacteria which holds the shade of the stain are known as gram-positive bacteria, whereas the bacteria which free the shade of the stain, that crystal violet shade stain, are known as gram-negative bacteria. Now this was a very easy definition to keep in mind. Next comes the peptidoglycan. This peptidoglycan, gram-positive bacteria mostly have thick multi-layer peptidoglycan. Positive one will have the thick, whereas the gram-negative bacteria mostly have thin or single layer. Here you can see I have drawn 1, 2 and 3. So it has a very thin line, whereas here you can see it has a very thick. Now this is your peptidoglycan. Next, Gram's reaction. So gram-positive bacteria maintain crystal violet color and stain purple or dark violet when it is washed with water and alcohol. They stay hued purple or blue with gram stain. The positive ones, which are positive, they'll have a purple or blue color. Whereas the gram-negative bacteria mostly decolorize to acknowledge counter stain. Okay, now what? So let's come with the shortest and easiest definition, which are as follows: characteristics, gram-negative bacteria and gram-positive. The first characteristic we must know is the cell wall structure, which I already showed, that the gram-negative will always be double layered. Here you can see it is double layered, whereas the gram-positive will be single layered, only one layer you can see here. So the next thing is peptidoglycan layer. Peptidoglycan layer will be thin. Here you can see it is very thin, which will be your 1 to 3 nanometer, whereas the peptidoglycan in gram positive will be thick, which will be your 20 to 80 nanometer. Next is your staining. As I mentioned previously, the negative will be pink or red, whereas the gram positive will be purple or blue. So the next one is your liposaccharides, which is your LPS. In gram negative, it will be present in outer membrane, whereas in gram positive it will be absent. So this was very easiest and shortest note regarding your this gram positive and gram. This was your shortest and easiest note regarding your gram positive and gram negative bacteria. I hope that it does help in exam. You gain good grades. Thanks for watching. Do share, subscribe, hit the bell icon. Thanks for watching. Bye bye.